Hello everyone. Today we are going to a new experiment named gram staining of microorganisms. The gram stain procedure distinguishes between gram positive and gram negative groups by coloring these cells red or violet. Here are the some apparatus or chemical or reagent requirements which is mentioned here like this. So let's start with a simple procedure of gram staining. First of all, we will have to take a clean glass slide, and after that we will have to put it in glass holder. We will take bacterial culture by using the inoculating loop, and after that we will have to smear it gently in the glass slide. As the smear is formed, we will have to heat fix it. We will have to fix the smear so that bacteria will fixed with the glass slide. It's a very important process, so we will have to do it very gently. Now bacteria is fixed with the glass slide. After that we will have to give some crystal violet on this slide by using droplets or fluently, and allow it to keep for some time so that the stain or crystal violet will bind to it the peptidoglycan in the cell wall. After 1 minute later, we will wash the slide by using a distilled water, gentle flow of distilled water, because we will have to remember one thing, that bacteria do not remove from the glass slide. Now we have washed this slide. Now we will use a gram iodine on the slide by using drop or drop wisely like this, and allow it to keep for 1 minute. After 1 minute we will have to wash the glass slide again by using this gentle flow of distilled water like this. After that we will have to wash the slide by 90% or 100% ethanol, and let the ethanol to keep on glass slide for 30 seconds. After that, wash the glass slide again by gentle flow of distilled water. Now it's the turn for secondary staining by using safranin. We will have to give safranin on glass slide dropwashly or fluently like this, and we will have to allow it to keep for 1 minute. Now we will have to again wash the slide by distilled water with a gentle flow like this. Now our slide is prepared. Now we will have to go for the observation of this slide by using microscope and by using the 45 or 100x zoom. This is a compound microscope. Here is a stage with a mirror. Here is a mirror also. Okay, and there is some screw also, and here is some lenses with various images of zoom, and we will have to keep the slide on the stage and we will have to adjust it through the screw and by setting the for 45x or 100x zoom. This is the microscopic view of this slide, and here we will see here the oval shaped bacteria here. Okay students, not more today yet. Thank you everyone, thank you all for being along the time with me. Stay safe and keep your family happy. Thank you all.